Today we are going to do a module on predictive modeling for food spoilage. This is the module 30 and it caters to a very niche area, that is predictive modeling. The entire module will be covered under the introductory portion. Then we will be talking about predictive models and their various types. We are also going to discuss the steps for development of these predictive microbial models. We are also going to talk about the available predictive model programs which are there. Finally, we will be also talking about the application of predictive models in food. We will be also comparing the predictive microbiology and the HESA program. We will be deliberating on the limitations of predictive microbial models, and in the end we will be concluding this module. When we talk about this predictive microbiology, it is very important to understand whenever we talk about food safety, we need to inactivate or we need to control the growth of spoilage and pathogenic organisms. Without controlling them, this is not possible to have safe food. Why? Because these are going to affect the shelf life of the food and also how much is the risk from eating contaminated foods. So, students, we should know what is predictive microbiology. This predictive microbiology is an applied branch of food microbiology and it describes the relationship between controlling parameters in food and a relationship between the spoilage and the pathogenic microorganisms. These responses, they are estimated and expressed by various mathematical equations and models. So, when we talk about predictive microbiology, we should also know what are the key features. Yes, the most important key feature is the mathematical expressions. What are these mathematical expressions? These equations, they describe the organism responses in food to various associated factors. These factors, they can be pH, acidity, temperature. Predictive microbiology is branch of microbiology which is integrating the traditional knowledge with the information system, stats, mathematics, and it is describing the behavior of microbes in the food. When we talk about the behavior of the microbes in food, we are talking about the factors such as the growth, survival, death or activities in microbes. And these activities are dependent upon certain intrinsic properties of the food. These properties can be the moisture content, the water activity, the pH, and side by side the extrinsic factors are also playing a very important role, and these extrinsic factors can be zeroed on to temperature, relative humidity, atmosphere, etc. Now, we have already understood that predictive microbiology is all about predictive models. So, what are these models and how do they help us? First thing, these models, they are capturing the changes in the microbial population as well as activities, and they can be estimated by the various changes which are happening in the product temperature, storage atmosphere, the pH changes, or change in the water amount or the water activity. These models are very useful for easy prediction of quality as well as safety of the foods. When we talk about the safety of foods, we are talking in a range of various types of conditions. Apart from doing that, these models, they also help us in comparing the various new product formulations, packaging and storage conditions. These predictive models are very helpful when we are designing or we are introducing new foods in the food industry or in the market. What are these predictive models? These models, they are going to predict the behavior of the microbes under conditions such as pH, temperature, salt, sugar changes. We are also going to take the responses of the microbes. These responses are then studied under specified set of conditions, and then the data which has been collected is analyzed and is expressed in the form of mathematical functions and evaluated to predict the new set of untested conditions. Most significant parameters are taken into the account for the formulation of these predictive models. When we talk about the predictive models, there can be various types of predictive models. The first category are the ones which are based on kinetics and probability. Usually, these are based on microbiological events occurring under set conditions. Then we have empirical and mechanical models. These are dependent upon various modeling approaches which the researcher is adopting. Then last, we have various types of primary, secondary and tertiary models where different types of variables are considered in formulating or constructing the models. The predictive microbial models, they are developed through various stages. The first stage can be the data which is collected. Most of the experiments are carried out under controlled conditions. Then this information is used for primary modeling. Based on the primary modeling results, we can also go into the secondary modeling. Once we have these kind of models which have been established, we go in for the product validation, and lastly, we can also use these models to predict further, and this is known as the tertiary modeling. Now we will be talking about the steps for the development of predictive microbial model. Like earlier I have said, the first step is generation of basic data sets under certain conditions. These conditions can be the growth conditions, they can be the survival patterns, or they can be the curves in specified combination of factors such as temperature, pH, salt, sugar concentration. When we are talking about this, most of this information is collected using liquid laboratory media and running the experiments in the lab. Then we go in for the development of primary models. In this, we are trying to fit the data by the sigmoid models, and we are trying to estimate the kinetic factors, factors such as time of lag and higher specific growth rate from the growth data graphs. We are also using suitable models which fit into this scheme. After this, we are moving on to the next stage, that is the development of secondary models. These models are helping us in the contribution of the control of the major factors on the kinetic factors such as the lag phase, the growth, the decline rate. After this, we move to the next stage, that is the model validation. What is this? In this, we compare the predicted values of the kinetic parameters to the product environment in challenge test. We are trying to simulate how this is going to work in an environment which is more realistic, which is more real time. Then we move into the development of models which are the tertiary models. In this case, we are trying to develop the software for the application of these validated models. When I talk about the predictive model, there are number of programs which are available in the market, and the one which is very critical and which is very popular is the pathogen modeling program, which has been given by USDA, ARIST, and is also available at their specific website. Then we have the next one, which is the growth predictor, and this has been given by UK, the country, and it is the successor of food micro model. Then we have another type of a forecast model which is known as the growth predictor forecast. It has been given by Camden and Chorleywood, UK. Then we have a specific model for the seafood spoilage, and it is known as the seafood spoilage predictor. This has been given by the Institute of Fishery Research, Denmark. Then we also have a very popular model which has been given by the University of Tasmania, Australia. This model is known as food spoilage predictor model. We also have predictive microbiology application softwares. This is only an indicative list. We have Combase, we have Simprevious, foodrisk.org, RIC, that is refrigeration index calculator. We also have Risk Ranger. Then we have specific programs for listeria. We also have fish shelf life prediction program, which is known as FSLP. Then we have process lethality determination spreadsheet, which has been given by the AMI Foundation, USA. Then we have shelf stability predictor. Then we also have one more program which is known as Thumb. This is only indicative list, but these are the most popular programs which are being used by the researchers across the globe. Now we know that we get number of models and softwares. We should be looking into the application of these models in foods. Why are these models required? They are faster, they are accurate, and the evaluation of the potential of the growth of microorganisms is very effective. This evaluation of the potential can be carried out under specified conditions, and the best thing is we need not be actually doing the test. This is more like a simulation of the real time conditions. Then we talk about these models. They can be applied for the development of new products and any change in the formulation before finalization. For pathogens, these models, they are very effective because the lag time is the most important parameter which determines the regrowth of the microorganisms. The time taken to produce toxins and cause food poisoning outbreaks also can be taken care of under specified conditions, and these specified conditions can be determined for toxigenic microorganisms such as Staphylococcus aureus B. cereus. We can also have specific models for fish, meat, fresh produce and yeast in fruits and drinks. These models can also be relevant to acidified foods, and we also have them easily available on the web. When we talk about the safety and stability of the acetic acid based mayonnaise and sausages, we can also have equations given in CIM SCEE code, which is SIMSE code. Pathogen predictions can be carried out using the Combase predictor system. The Combase system contains growth and thermal death models. These thermal death models predict the log reductions of an organism at a specified temperature. When we talk about the application of predictive models in food, we cannot ignore the SSP model, which is the seafood spoilage predictor. This software has been developed specifically to predict the effect of constant and fluctuating temperature conditions on the shelf life of the sea products. The next model, which is very important, is the food spoilage predictor. This is FSP. This model can be used along with several other predictive models which are available for the prediction of growth of psychotolerant pseudomonas in fresh and aerobically stored fish. The model which is very important and which is being regularly used in USA is Pathogen Modeling Program, which is PMP. This program includes 13 models for growth and survival of various pathogens. It also has 9 models for inactivation of pathogenic bacteria. The other model which is important is the food micro model, which has 23 growth and survival models and 7 models for heat inactivation of pathogens and various microorganisms which are capable of causing foodborne diseases. We have models in PMP and food micromodel which study or which predict the effect of wide range of controlling factors such as the salt concentration, the vacuum, the modification of the product atmosphere. It is very important because we are getting new packaging systems in which we are targeting the modification of the atmosphere of that particular pack. Now when we talk about the selected models, it's very important to understand how are they being constructed. They are usually made by using microbiological media as a means of obtaining information of microbial populations, how these microbial populations are going to behave in foods. The user must ensure their validation by using their own extensive experience along with conducting a series of various experiments under environmental setting. Means you can see the growth of a particular microorganism at different pH parameters, at different salt concentration and even at different acid or sugar concentration. We can see how the pathogen modeling program PMP works online. It's a very simple program in which we can talk about various types of models which are dealing with bacteria or yeast or molds. We can also pick up the models for specific food products. If you see here, we have selected a model for the bacteria, and once the model for the bacteria has been selected, we get a range of microorganisms which can be chosen from under the list of bacteria. You can see on the screen that we have Aeromonas hydrophila. We can also talk about Bacillus cereus, Salmonella, Escherichia coli, Staphylococcus aureus. All these bacteria can be studied in single food or in separate environmental conditions. When we see this model, we can also go and select a model which is for a specific pathogen. For example, you can see on the screen we have selected a model which is for Clostridium botulinum, and the model has been taken in case of the broth culture, and once we have the broth culture we can use various linear models, and you can see these are reflected in the form of the graph, which is the line graph on the left side, which tells us how this pathogen is going to behave in the broth under different conditions. This is another very popular model which is food spoilage and safety predictor, which is FSSP. Various versions are available in the market. On the screen you can see the version 4. This software predicts the growth of spoilage and pathogenic microorganisms in food. The major objective is to develop a user-friendly software to predict the effect of different types of fluctuating temperature conditions and also how the product is going to behave. The software includes various types of components which are like the 4 product specifications depending upon the rate of spoilage models. We can also have a generic model to predict the microbial growth with the shelf life. FSSP is a model which can be used by the users by incorporating their own data on the growth of microorganisms. Now we will be discussing 2 models which have found utility in various types of predictive modeling studies. These softwares are the square root type model, which is used again to predict how a pathogen or a microorganism is going to behave. We can pick up various sets of conditions like temperature, the percent CO2, we can also take how much is the cell density, we can also take what is the water activity, and all these parameters can be used to predict the shelf life of the food product. On the other hand, we have one more model which is our SS model, which is equally a user-friendly model which can be used for predicting various types of growths, and we can select the temperature of choice. We can also predict the equivalent shelf life at that temperature. This model is very helpful in predicting the shelf life of a food product under fluctuating temperature conditions. Now you can visualize a screen where we are seeing the relative rate of spoilage. Various types of models are adopted and we can forecast how the microorganism is going to grow under different sets of conditions, and depending upon the projections which are received by this model, we can predict the shelf life and side by side also the conditions which are going to keep the food safe. You see here a generic model which is predicting the growth of Listeria monocytogenes. In this case, we can modify the various temperatures. We can also modify the various types of components which are being added. We can also talk about like organic acid. We can talk about how much will be the relative time of growth, and we can predict the growth. This growth model really helps us to control the growth of Listeria monocytogen, which is a very important pathogen and it is a threat to food safety. When we are talking about the growth models, these models not only predict the growth of pathogens, we can make use of these models for the growth of useful microorganisms such as lactic acid bacteria. We can also predict how a microorganism is going to grow over a period of time, and if you see in this graph, you will see a plateau which has come after the growth. We see that the microorganism has undergone an exponential growth and then it has reached a stationary phase, which is due to the accumulation of the acid, that is the lactic acid being produced by this microorganism, which is acting as inhibitory due to its concentrations at the later stage of its growth. Predictive microbiology is strongly interlinked with HESA because HESA is a system which predicts the potential food safety problems, and we can simulate those models in the predictive microbiology, and we can always see that the levels should be kept within the control, and it will also help us in risk assessment and predicting the shelf life. But although I say that both the things are interlinked, that is HESA and predictive microbiology, but still there is a difference between the two because predictive microbiology identifies the microbes of concern, but HESA is only focusing on the potential hazards. Then when we talk about predictive microbiology, we are talking about ecology of the microorganism in which it is going to grow, whereas in case of HESA, we are only talking about the CCPs, the critical control points. In case of predictive microbiology, we are comparing a preset control specifications. These can be taken, these can be rejected. But in case of HESA, we are talking about the control criteria and the methods where the control has been achieved or it should be achieved as and when required. When we talk about the predictive microbiology, we see that we are trying to incorporate the available information into the monitoring systems, whereas in case of HESA, we need to establish and implement monitoring procedures to a system where there should be no deviations as per the regulatory guidelines. Now, after knowing a number of models which can be suitably adapted for various types of foods like seafoods, meat, fresh fruits and vegetables, we can see that predictive models, they have implications or they have uses in multiple sectors. But yes, these predictive microbial models, they also have their limitations. For example, they cannot be extrapolated outside the range of information data which we have got. These are useful only in case of development of models which can be of temperature, moisture, etc. Prediction outside the range is very tough and it cannot be very precise. These predictive models are usually developed based on the data which is generated using laboratory media. These cannot be extrapolated in food systems, and even after they are validated in foods, they might not have a widespread application in the diverse food industries. Why? Because microorganisms are going to behave differently under different products and under different sets of conditions. Models generated under static environments cannot be effectively be applicable in fluctuating conditions. These fluctuating conditions are usually a norm in case of food products because there can be a change in the environmental conditions such as the temperature during the transportation or during the storage, during the retail or other related factors. There is a variability in microbial populations, physiology, genetic makeup which also limits the application of these models. These models have to be more precise if they have to be implicated or they have to be used in different types of food systems. Now coming over to the summary of this module, which you might find little bit tough as most of the studies are based on the online computer models. The predictive models are an alternative to classical assessment of microbes. These evaluate the quality as well as the safety of the foods in a faster, easier and cheaper manner. They are trying to integrate the knowledge of the ecology of microbes in food as explained, and they can also evaluate the development of microbes under the processing, storage and distribution conditions. Various softwares can be used to predict the shelf life, and still there is a scope of lots of work to be done in this particular area. Thank you.